Good evening to everyone present here. Warm welcome to all. In connection with Dhanvantari Jayanti, Department of Rog Nidana of Ayurveda College and Hospital, Coimbatore, and IAS Ayush has been organizing a webinar series, and today marks the day 4 of it. The topic of today's session is isolation of pathogenic bacteria from urine, stool, and sputum. We have Dr. Shashirekha ma'am. Welcome, ma'am. Dr. Shashirekha ma'am is a well-known microbiologist and currently rendering her service in Roganidana department in Government Science College, Hassan. Ma'am has over 7 years of teaching experience. Ma'am has completed her MSc and MPhil studies and is also on her way to complete her PhD studies. She has co-guided many students pursuing their PG studies in Ayurveda and Masters in Microbiology. Ma'am has a diverse research experience wherein she has conducted research on topics like preparation of antimicrobial drugs from Ayurvedic drugs, antimicrobial action of Dupana with respect to airborne microbes in indoor environment of central hospitals, and development and validation of polyherbal urine and its in vitro study on organisms using auditors externa, etc. Ma'am has also published several research papers in national and international journals on topics like antimicrobial activity of herbomineral preparations on multi-drug resistant bacterial isolation from humans. She has also conducted study on in vitro study of endophytic bacteria, bacillus cereus, from tulsi weed against Klebsiella pneumoniae by urine culture and sensitivity in Physica and the list just goes on and on. Your journey is not only appreciable but also motivates us to reach great heights. Ma'am. I now request ma'am to share her knowledge and experience to be a guiding light for these students. Over to you, ma'am.
Am I audible?
Yes, ma'am.
Hello? Neha?
Yes, ma'am. You are audible. Yes, ma'am.
Sneha? Am I
Yes,
audible?
ma'am. You are audible. Yes, ma'am.
Okay. Can I start the presentation?
Yes, ma'am. Sure. Ma'am.
Yeah. Good evening to one and all. Myself, Dr. Shashirekha, a microbiologist, Government Science College, Hassan. Today, my topic is on isolation of pathogenic bacteria from urine, stool, and also sputum. So let's start with the introduction. That is a introduction to medical microbiology. So, medical microbiology is a study that deals with the interaction between the parasite and also host. The host, it may be the plant, human beings, and also animals. And these specific microorganisms that might be useful and also harmful for both plants, human beings, and also animals. If it is a useful, it might be example for in a human beings. It will be gut flora, which will be having the production of vitamin K in a very little amount. It will be done by E. coli, the gut flora. In the same time, the harmful effect for the human beings, the same E. coli, which will be causing the UTI, right? That is a harmful effect. So the interaction in which the microbes are benefited and also humans are harmed. That is a harmful effect from the microorganisms. That mechanism is known as a parasitism. So, The medical microbiology is a branch of medical science concerned with the prevention, diagnosis, treatment of infectious diseases. There are four kinds of microorganisms that we are all know, that bacteria, fungi, parasites, viruses, and one type of infectious proteins called prions. Right? Uh, Coming to the medical microbiology laboratory, what we will do in a medical microbiology laboratory, it is a place when the diagnosis of infectious microorganisms takes place, the identification of best treatment option for the infection, and the monitoring of antibiotic resistance. And it is also includes testing for how well a patient is responding to a treatment of infection. So that is what we are going to do in a medical microbiology laboratory. So we'll see what and all the instruments we can see in a general medical microbiology laboratory. So we can see the pH meter, spectrophotometer, centrifuge, a water bath, rotatory shaker, laminar airflow, incubator, digital weighing balance, centrifuge, magnetic stirrer, microscopes, culture medias, and also inoculation loops. These are the minimum basic things which we need in a microbiology laboratory, right? And also, along with that, the instruments which we will use, the basic concept of medical
Hello, ma'am.
microbiology laboratory
Sorry to interrupt,
is
ma'am.
with the use of artificial media, by providing the artificial media to
Sorry to
the
interrupt, ma'am.
microorganism. Yeah, tell me. Yes, tell me.
Moving. The slides aren't moving, ma'am. Yes, now it is moving.
Can I use with this screen only?
Yes,
Because if I
ma'am.
am making a larger one, it may not move. Now it is moving.
Yes, ma'am. This is fine, ma'am.
Okay.
Yes.
Okay. Along with the instruments, medical microbiology lab, which will be having all these instruments, along with the instruments, we have a different culture medias. Culture medias are the artificial medias which will be providing the nutrition for the growth of microorganisms. Any type of microorganism, bacteria, fungi, microorganisms, they need some amount of nutrition for their growth, nutrition and also substrate for the growth, right? So based on the consistency of the culture media, medias have been classified into three types. Basically, the solid media, you can see here in the picture, this is a plate which is containing the solid media that will provide a substrate for the growth of microorganisms, and also the area of this solid media will be providing more area for the growth of microorganisms, right? So this is a solid media. Based on the consistency of the medias have been classified into three types: solid media, liquid media. Second one is liquid media. You can see the test tube which is containing the liquid, that is a nutrient. For example, nutrient broth. Nutrient broth is a liquid media which will provide the nutrition for the growth of microorganisms. Here, the solid media, which will be, this is the plate where you can see the red one, solid media. So this is the plate. It is a solidified media. That means with the media, we are providing the agar-agar. Agar-agar is a solidifying agent that will make this media into solid texture, right? So this is a solid media. Liquid media is the be there in this liquid media. So it will be in a liquid consistency, right? And this next one is semi-solid media. Semi-solid media means it won't be solid, it won't be liquid, right? It will be a medium solid in texture, and it will be a jelly like consistency to provide the consistency. I'll be telling, is it audible? Is it audible? Is it audible?
You're audible, ma'am.
Am I audible, Sneha?
Yes, ma'am, you're audible, ma'am.
Yeah, okay. Sir, I'll use
Mhm.
this small screen only, sir. Slides are not visible, it seems.
Yeah, okay, no problem, ma'am. This is okay, this is fine.
Yeah. Yeah, okay. So coming on to the slides, so we have a three medias based on the texture of the media. We have a three types of media, that is a solid media, liquid media, and also semi-solid media, right? So I'll be discussing in the further slides what and all the use of where and all what type of microorganisms we can grow in this types of media, right? So the liquid media, for examples for liquid media, we have a peptone water, nutrient broth, glucose broth, and also Robertson cooked meat media. I'll tell you, and also we have a selenite F broth, alkaline peptone water, thioglycolate broth, and tetrathionate broth. So these are all the different types of medias for the different types of microorganisms, right? The peptone water, for example, it might be using for the cultivation of basic microorganisms. But Robertson cooked meat media, peptone water, nutrient broth, glucose broth, this will be used for aerobic bacteria, cultivation of aerobic bacteria. But this Robertson cooked meat media, alkaline peptone water, these will be used for anaerobic bacteria. So there is a classification. Again, there will be a classification based on the requirement of oxygen for the growth of organisms, right? Based on that, there will be a different types of culture medias will be there. Whether it is a liquid media, solid media, there will be different types of culture medias will be there based on the requirement of microorganisms, right? We have a liquid media, peptone water, peptone water, nutrient broth, glucose broth, and also Robertson cooked media, along with the alkaline peptone water, thioglycolate broth, tetrathionate broth, and also selenite F broth, right? So next is the solid media. Solid media, we have nutrient agar, blood agar, chocolate agar, bile salt agar, lower flush serum slopes, TCBS agar, McConkey agar, Dorset egg medium. So these will be having a different compositions. Like agar will be differs, and also along with the bile salt, sodium citrate, sucrose, ferric citrate. So the every single agar will be having its own composition for the growth of a specific media. For example, this TCBS agar is a agar which will be meant for Vibrio cholerae, and also this one chocolate agar, blood agar, and chocolate agar. This will be gives the growth for the nutrition for the growth of gram positive bacteria. Gram positive bacteria will used to grow on this blood agar and also chocolate agar. Nutrient agar is a basic media where all types of organisms can grow, like gram positive and also gram negative organisms can grow on this nutrient agar. And also McConkey agar. When coming to the McConkey agar, some of the organisms, gram negative organisms like E. coli, Pseudomonas, Proteus, all these organisms will can grow on this McConkey agar. And also this McConkey agar is also known as a selective and also differential media for the growth of bacteria. So differential media, I'll tell you in the next slides. So coming to this next one, semi-solid media, we have The semi-solid media you can see in this test tubes. So it will be very jelly-like consistency. And here you can see this is the right side is a media without inoculation, and left side you can see here, left side, left test tube you can see the test tube is fully cleared, and middle you can see one growth up to the end. There will be a stabbing has been done, and there you can see the growth of microorganisms. So this is the media which will be used to see the motility of organisms, the organisms which are having a flagella, and they will move inside the liquid media. If you are providing a liquid media, it is very easy to easy to show the motility inside the liquid media. But on a solid media, the motility will be declined due to because of the substrate will be there, where the substrate, the organisms will be adhered in that media. But if you want to know whether the organism is flagella, whether the organism is having a motility, then you have to do this semi-solid media. You use the semi-solid media, and you can test the motility of the organism. Then you can definitely know whether the organism is having a flagella or not. For that specific requirement, we will use this semi-solid medias to test the motility of organisms. Okay. So coming to what and all the things which we will do in a routine microbiology laboratory. That is a routine work steps. What and all the work steps we will follow in a microbiology laboratory, we will see. Okay. The microbiology laboratory, the lab practice involves the following steps. That is a first the reception of samples. You all will be knowing the diagnostic lab will be there. If the patient is having any infections or if he is suffering from any infectious diseases, the physician will prescribe some culture and sensitivity test, right? So for that, what the patient will do, the patient will collect the sample, and they will be reporting, or they will be giving to a laboratory person. So that is a reception of sample. The universal sample collection bottles will be there in a laboratory. For the specific specimens, that is a urine sample, stool sample, sputum sample, and also pus sample, there will be a specific collection bottles will be there. So that specific bottles will be given to the patient. The sample will be collected from the patient, and it will be labeled with the name, date, age, right, and the sample. So that is a reception of sample, and the sample will be cultivated on a specific culture media. Then microbes will be identified based on their also antibiotic sensitivity, right? So, what is the purpose of for doing this culture and sensitivity? So, what we can do is we can guide the clinician in selecting the best drug for the individual patient. The specific drug, if you are giving one drug to the 10 patients, it may not work out, because the every single patient is an individual patient. The body will be working differently for individual patients, right? So for the specific person, for the specific body conditions, they have to clinicians has to give the best drug for the individual patient. So if you are doing a culture and sensitivity, you will get an one idea which drug will be best for your infection, and also to control the inappropriate use of drug in a clinical practice, to accumulate epidemiological information on the resistance of microorganisms for public health importance in a community, and also to reveal the changing trends in local isolates to overcome microbial drug resistance, right? So next we will go for, so we have understood why we are doing culture and sensitivity. So we will go for the first, first method, that is the urine culture and sensitivity. The sample will be the patient, if patient is suffering from a UTI or any fever or something. So a physician will be prescribing for a urine culture and sensitivity, right? So in the diagnostic lab, the samples will be arrived with along with the patient name, age, date of collection, time of collection. Everything will be written on this. So you can see here, this is a universal urine sample collection bottle. So you can see this one, this bottle. Am I audible? Sneha, am I audible?
Yes, ma'am, you are audible, ma'am.
Yeah, okay. So, this is the universal sample collection bottle where we can collect the sample of urine sample. The early morning urine sample will be preferably used for isolation of microorganisms. Early morning, midstream urine will be collected. So after collection of the samples will be sent to the laboratory. Here you can see the centrifugation machine, which is holding the, this is a centrifuge tubes. You can see here the two tubes are there. This is a centrifuge tubes. The sample will be put it into a centrifuge tubes that will be containing 5 to 10 ml of centrifuge tubes only. The sample will be poured and the sample will be centrifuged at a 2500 rpm for 2 to 3 minutes, right? So after that, you can see here in test tubes, which will be having a pre and also post samples. You can see here the tubes, centrifuge tubes. In a first sample, you can see the turbidity of that urine sample, right? The turbidity which shows the sediments which are collected in the second photo picture. So all the samples, all the samples which will be containing the epithelial cells, cast formation, pus cells, or microbial load, which will be equally distributed in a first test tube. You can see here, this is a turbid urine sample test tube. So which will be having a evenly distributed turbidity, right? So the next, after doing a centrifugation, there will be a settled sediments, you can see here. Settled sediments you can see here, and the urine will become clear. And this clear supernatant will be discarded, or it can be used for the protein estimation. And the below, the sediment will be used for culture and sensitivity, culturing of microorganisms, and also for the microscopic observation, what and all the particles will be there in a microscopy. To see that, we will use this sedimentation. Okay, so what we will do next is, so this sedimentation, we are going to take a one loop full of sedimentation on a glass slide. We are going to take a one drop of sedimentation. Here you can see the picture. The first is a urine sample. After centrifugation, how the sediments will be settled here, and that sediments will be taken for a microscopic observation. The microscopic observation will be done by doing the taking the one drop of sedimentation onto the glass slide. Glass slide mele one drop of sedimentation you have to put, and you have to cover with the cover slip, right? So after doing this procedure, we have to go for microscopic observation. First, we have to focus in a 10x, then we have to go for a 40x for the larger vision. So under the microscopy, what and all the things we can see? So here in the picture, you can see the urine microscopic observations. That is a budding yeast, pus cells, motility of bacteria, RBC cells, different types of tubular squamous epithelial cells, and also spermatozoas, RBCs, RBC cast formation, WBC cast formation, epithelial cast formation. You can see. Okay, so this indications. So RBCs and all you can see in a hematuria, right? Vascular damage. You can see RBCs. White blood cells will be visible in a pyuria, the pyuriaದಲ್ಲಿ, and also nephritis. Renal transplant rejection cell. You can see white blood cells, and also squamous epithelial cells. Increased indication contamination of urine with the vaginal fluid. You can see this, and also you can see the transitional epithelial cells in a viral infection and also malignancies. Then I'm talking about this epithelial cells. There will be a different types of epithelial cells, transitional, squamous, and also renal epithelial cells. So there will be a specific indications will be there. Okay, so and also the bacterial cells you can see here. This is a bacterial cells we can see in a urinary tract infection. Urinary tract infection only we can see all these things, right? And moving on to the next one, that is crystals. Okay, the crystals you can see here the different types of crystals. Different types of crystals. The uric acid crystals, which can be seen in the tumors, lysis syndromes, and calcium phosphate crystals. You can see here calcium phosphate, where it is not moving. Calcium phosphate, calcium phosphate crystals, not a suggestive for any specific systemic diseases, but magnesium, ammonium phosphates. It is seen in a UTI by urease producing organisms. Calcium oxalate, calcium oxalate crystals, that will be calcium oxalate monohydrate, that is an seen in ethylene glycol indigestion, and also cystine crystals, that is diagnostic for cystinuria. Cystinuria. There will be a specific crystals will be indicating a specific diseases in a specific individuals, right? So next is the culture techniques. Culture techniques. After receiving this sample, we have did a microscopy, right? After receiving this sample, we have did a microscopy. After observation, we have got a urine microscopy, budding yeast, fossils, bacterial cells, RBCs, different types of epithelial cells, spermatozoas, and also different types of gas formation, and also crystals, right? So after doing this, we have to identify which type of bacteria is causing the infection, right? So for that, what we have to do, we have a specialized medias for the growth of specific bacteria, right? So the culture techniques we will employ for the cultivation or for the isolation of bacteria which is causing or which is infective in a individuals. So this is the sample we have got, that is a sedimentation. Here in the picture you can see this is a sample bottle which is having a sedimentation. So that sedimentation which will be placed on this culture media. So culture media, we are going to place one drop, one loop full of inoculum. The inoculum is nothing but the sample. The sample, that is a sedimentation, that is known as an inoculum. That inoculum, one loop full of inoculum, the inoculation loop. In a below picture, you can see this is the long inoculation loop is there. The person is holding in his hand. That is a loop. Here you can see the loop, one loop. That one loop full of sample will be taken on a culture media, and we have to do a culture streaking. Streaking. It might be a lawn culture. It might be a zigzag. It might be no parallel. Parallel streaking. This is a parallel streaking. You can see in this third picture. This is a parallel streaking, and this is the streak zigzag streaking. Okay, why we will employ all these different types of culture techniques is to isolate the pure single isolated colony. We can see here after doing this zigzag, here you can see in the second picture. So the primary streaking, this is the primary streaking. There you can see the huge number of microorganisms. The huge number of microorganisms will be present in the primary streaking. From that primary streaking, we have to take a secondary streaking. This is a zigzag streaking. Here you can see in the end of this streaking, the number of colonies will be less and it is larger in size. You can read the colony morphology of the particular organism. Or for that, we will go for streaking techniques. Different streaking techniques will be employed for that purpose. Why? We need to study the different colony morphology, or we have to study the specific individual organism, what type of organism it is, which kind of organism it is, we can study by doing this culture techniques. So this is a sample, or one loopful of sample will be taken on a plate and we are going to streak it with the help of sterile inoculation loop. After streaking, we are going to place this plate in a incubator for 24 to 48 hours. After putting a plate in an incubator, we are going to incubate this plate for 24 to 48 hours of incubation. After the 24 to 48 hours of incubation, the organisms will be grown on this plate like this. The zigzag streaking or parallel streaking, whatever the streaking patterns you are doing, or wherever the streaking has been done, on that area the organisms will grow after 24 to 48 hours of incubation. This is a culture techniques. Okay, so this culture technique will be same for all urine, stool, sputum. The media will be different. For the urine, we are using McConkey agar, blood agar, and also nutrient agar also can be used. The specific medias will be there. The nutrient agar, blood agar, and also McConkey agar. So these medias will be used for the cultivation of urine culture. The culture techniques, what I said, the zigzag staking, parallel staking, and the inoculation of sample. This sample will be differ. This is a urine sample. The next technique will here it will be a stool sample. Next technique it will be a sputum sample, the pus sample, like that it will be. The only the difference is the urine sample will be sedimented, not all the samples. Stool samples, pus sample, and also sputum sample will be inoculated as it is onto the culture media, but the urine sample will be centrifuged for the sediments. Okay, so the same technique will be followed for urine, stool, sputum, and also pus samples. The culture techniques will be same for streaking, incubation, inoculation, streaking, and also incubation will be same for urine, stool, and also sputum cultures. Okay, moving on to the next slide. After this, streaking, we have to see for the colony growth. So as I said, the urine sample will be cultured on a McConkey agar, right? So earlier I told the McConkey agar is a selective and also differential media. So what is the differential media? So the McConkey agar will be having a lactose sugar. So that will be in a pink color. The media will be in a pink color. The organisms which are utilizing fermenting, the organisms which are fermenting lactose, which will be a pink color colonies. Here you can see the second plate, that is a pink color colonies. The next to that plate, the plate was earlier the pink color, but after the incubation the plate has been turned into a yellow color. Why? Because this organism has not fermented the lactose. Okay, so the organism we can differentiate whether it is a NLF or LF. LF is a lactose fermenter and LF is a non-lactose fermenter on a McConkey media. The organisms we can differentiate on this culture media. McConkey media is also used as a differential media to differentiate whether the organism is a lactose fermenting or non-lactose fermenting. Lactose fermenting organisms will produce this pink color on a culture plate, but the non-lactose fermenting colonies won't show pink color colonies. They will be pale color colonies. Okay, That is the use of this Mekong data. Okay. Next one is after cultivation. After cultivation, we are going to identify the organism. Not only is it not only sufficient for only the growth of organism, but we have to identify what kind of organism it is. So for to know that, we have to follow the common procedures. That is a we have to study the colony morphology of a specific organism. This is the organism. We have to check for the colony morphology. The single organisms, you can see here the streaking, the last streaking, you can see the single isolated colonies, right? So this isolated colonies, the single colony, you have to check for the colony morphology. That means elevation, forms, margin, surface, size, optical features, consistency, chromogenesis, smell, blood hemolysis. It is considered as a preliminary step. Okay, the colony morphology is a preliminary step where we are going to see the elevation. This is considered, this is a single isolated colony. This is the elevation, how it is made, the entire idea, elevated idea, this circinate idea, filamentous idea. So we have to check for all the colony morphologies. Then we have to go for a staining methods, whether we can use a gram staining based on the organisms, what kind of organisms we are going to stain. We can use a Z-Nielsen staining method also. And also the biochemical test, which will helps us which species the organism is. The biochemical test may include IMBIC test, catalase, coagulase, oxidase test we can see, and also molecular methods, PCR methods, ELISA methods also we can use, but that is a less common than above mentioned protocols. That is a colony morphology, staining, and biochemical, which will be feasible. Okay, than the molecular methods. So what I said is a colony morphology. So you can see here the four different types of bacteria which are showing the same nutrient agar plate. This is the same nutrient agar plate which we can see different types of bacteria. The first one is Bacillus subtilis, which you can see the dry, circular, dry, circinate, irregular, right? And also creamish colonies on this Bacillus subtilis plate. And here the Proteus vulgaris, Staphylococcus aureus, Staphylococcus pyogenes. So you can see by seeing the colony morphology itself, you can tell the organism genus level. Genus level organism identification can be done by seeing the colony, by reading the colony morphology. Okay, so colony morphology, you can see here the size will be measured with the scale only. You can use the scale. By the scale you can check whether it is a how much diameter it is. And the color of the colony, you can see here the different types of colors. You can see the pink color, pale yellow color, cream color, right? Greenish tinge is there, golden brown color is there, white creamy color is there, right? So that is a different text, different colors, right? And next one is a texture. Texture, you can see the texture is dry, smooth, viscous, right? And also mucoid. See here, you can see here the mucoidiness. See this one, non-lactose fermenting colonies, you can see this is a mucoidy texture. And here you can see the Bacillus subtilis, it is a dry one, right? So that is based on the different textures of colonies, you can identify the genes. The elevation from above, from the plate, if you are watching the plate from the above, you can see the single colony, whether it is a flat, raised, convex, ambonate, critiforms, right? So different types of colony elevations you can see. And the forms, the forms will be identified with the edges only. The whole form, how the what the shape it is. The one single colony, whether it is a one single colony which is having a round shape or irregular shape. See, this picture will be showing this non-lactose fermenting colony. You can see here the single isolated colony, which will be having an entire round it is, right? That is a round. But here in a Bacillus subtilis, you can see the irregular colonies, right? It is a irregular colonies. By identifying that, you can see the forms. That is a filamentous or rhizoids. So you can see the forms with the single colonies, and also margins. You have to see for the edges of the colony, single edges of the colony. If the organism is having a round form, then definitely the edges will be entire, right? So if the irregular form, if the organism is having the irregular form, there will be a low weight, right? Like that, the different forms and also margins will be there in a colony morphology study. So you can, by seeing this picture, you can easily identify how the entire form, critiform, right, ambonate form, raised and also concave, margin will be low weight, curled, undulate, right, glistening, circular, irregular form, and also glistening, and also color will be white. This is the yellow color, rhizoids, and also this is a filament, right? So that is a different colony morphology structures we can see with the organism. So after doing this, identification, we have did a colony morphology by seeing the form, elevation, margin, surface, size, optical consistency, and also chromogenesis. Smell. When coming to the smell, we can see this lactose fermenting. We can see definitely the curd, how curd smells like that. The lactose is a sugar that will be fermented by the organism that will be giving the lactose, that is a curdy smell. If you are smelling this, the if you are opening, just smelling this, pass your hand along with the plate, that will give you curdy smell when it comes to the E. coli. Okay, and also when it comes to the budding yeast, that is a Candida albicans or a budding yeast, that will give you a fermented smell like a dosa batter, alcoholic smell. Okay, why? Because it will be fermenting the media which is present in the plate, right? And also the which one, Staphylococcus pseudomonas will be giving you a soxy smell. We will call it as soxy smell, right? Like that, there will be a different types of organisms will be producing different smell. By that also we can identify the what kind of bacteria it is. So next is moving on to, after the colony morphology, we have to study the gram staining. That is a staining technique, as I said earlier, the in a microbe identification only. So we have did a colony morphology first, then we are moving on to staining of bacteria. So this is the bacteria. So from this is a single isolated bacteria. You are going to pick it for doing the staining method. So how, let's see how the staining will be done. Okay. So the gram staining is a procedure which is a differential, specialized staining method. The differential, why we will call it as a differential, is we are going to differentiate the gram positive and also gram negative bacteria. Okay, so this is the staining method where we will having four different types of staining reagents. The first one is a primary stain, crystal violet. Second one is a moderate, grams iodine. Third one is a decolorizing agent, alcohol. And the fourth one is counter stain, that is a saffron. Okay. So first what we have to do is we have to take a colony, one selected colony on a glass slide, clean grease-free glass slide. On that we have to put one drop of water along with the colony. We have to smear, a thin smear will be prepared with the use of inoculation loop. Okay, we have to prepare the inoculum, that is a smear. We have to prepare a smear on a clean glass slide of the bacteria. So then we have to air dry it. After drying, you have to you have to flood the smear with the crystal violet. We have to flood the smear with the crystal violet. Crystal violet will be violet or blue color. The color of the stain will be a blue color. Here you can see the picture of gram staining. The first one is the first procedure is we have to smear the bacterial colonies, bacterial bacterial inoculum on a clean glass slide, air dry it, then you have to pour the crystal violet on the smear, leave it for a 30 seconds. Leave it for 30 seconds. After 30 seconds, we have to wash it under the water, then add grams iodine. Grams iodine, that will be a brown color. Grams iodine will be moderate, that is a brown color, that will be left on a smear for 30 seconds. Again 30 seconds we have to leave this gram staining. I have to grams iodine moderate for 30 seconds. After 30 seconds again we have to pour, wash out the smear, wash out the color, then add decolorizing agent, that is alcohol. Alcohol is a decolorizing agent which will remove the excess of stains which are present on a smear. Okay. Then again what we have to do is we have to add saffron, alcohol, which should be left for 15 seconds. The first one is crystal violet, that will be 30 seconds, iodine for 30 seconds, alcohol for 15 seconds, and last one, the counter stain, the saffron, that should be poured on a smear and left for the 30 seconds. After the 30 seconds, wash it under the water, then leave the preparation for drying and also observe under the microscopy. So the microscopic observation, if you are observing under the microscopy, here you can see the down picture, both the pictures, gram-negative and also gram-positive bacteria. If the gram-positive bacteria, the organism is a gram-positive bacteria, that will be taken out. The blue color, the primary stain which we have treated, that is a primary stain, that is a crystal violet. That will be a violet or blue in color, that will be taken up with the gram-positive bacteria. And the if the organism is gram negative, the organisms will taken up pink color stain. Why? Because the bacteria, the cell wall of the bacteria will be having a peptidoglycan layer. Both gram positive and also gram negative bacteria will be having the peptidoglycan layer, but it will be thick in gram positive cell wall. Gram positive cell wall will be having a thick peptidoglycan cell wall compared to gram negative. So that peptidoglycan will be very thick, that cannot be reached by the decolorizing agent. Even after treating with the decolorizing agent, the lipids which will be present in the peptidoglycan, that will be holding this primary stain. What you have given, the primary stain, the blue color, that will be held by, tightly held by the peptidoglycan layer. Even after treating the decolorizing agent also, the cell wall won't leave that primary stain. Okay, so that's why the primary stain will be taken up by the gram positive cell wall. But this gram negative cell wall will be having a peptidoglycan layer, but it will be very weak. After treating with the decolorizing agent, all the color will be came out, but after treating the saffron, again the cells will be taken up the pink color. You can see here the first two pictures are blue color, that is a primary stain, crystal violet and also moderate. Then after treating with the decolorizing agent, the colonies will be colorless. The gram negative organisms will be colorless, but here you can see the gram positive organisms are blue color. When you have treated with the saffron, the colorless transparent organisms will taken up with the counter stain. That is the difference why the gram positive will taken blue color, gram negative will be taken up with the pink color. Okay. So this is the primary staining. By this staining, we will get to know that whether the organism is a gram positive or gram negative. This will be very much helpful in selecting the antibiotics also for the individuals. The next one is the biochemical test. The biochemical test will be a very long list of biochemical tests we have to do for specific organisms. Specifically, if you're doing for the Escherichia coli or Enterobacteriaceae members, we can select the IMVIC series. That is a indole methyl red Vogus prosperous citrate utilization test, and also we can do the sugar fermentation, indole production, methyl red test, then urease test, catalase production, oxidase reaction, and also PSA, that is a triple sugar iron media test. These different types of biochemical tests also we can conduct for identification of species. The genus level we can identify roughly with the colony morphology, but if you want to identify the specific species of organisms, you have to go for the biochemical test like INVIC and also urease, catalase, oxidase, and also TSI media test also we have to do. Okay. So this is after after we are studying the colony morphology, doing gram staining, doing biochemical test, we will be knowing that what kind of bacteria, what type of bacteria it is. Okay, so based on that, we have to select the specific antibiotics for the to get to know what kind of which kind of drug will be working out for a specific bacteria, right? So for that we have to do the sensitivity. Culture, up to now we have did a culture and also biochemical test. After that what we have to do is we have to select a specific antibiotics for a specific bacteria. For that what we have to do is a sensitivity is where we have where we can use the two types of medias. That is a specialized media Muller Hinton agar also we can use for lawn culture, and also the nutrient agar also we can use for sensitivity. So here you can see this is the first picture. You can see this is the media which have been swabbed, inoculum have been swabbed. This is a lawn culture. The lawn culture is nothing but the plate, whole plate will be swabbed with the inoculum. Inoculum is nothing but the old colony where we have grown. See, this is a technique what we have done. This is a sample, urine sample, which we have inoculated on a plate. The organisms have been grown. This grown organisms we have undergone with the gram staining, biochemical reactions, colony morphology we have done, gram staining, IMBIC reactions also we have done. The same, these colonies, this colonies will be taken for a culture. Again, lawn culture on the specific media. Either we can choose a nutrient agar or Muller Hinton agar for a sensitivity. Okay, the plate will be swabbed, whole plate will be swabbed with the inoculum. Inoculum is nothing but a bacterial sample. Okay, so the plate will be swabbed with the bacteria. Then we have to select the specific media, whether we are using a gram positive or gram negative bacteria we are treating. We are based on that the antibiotic disc will be selected. See here in the second picture we have a specific antibiotic disc. Antibiotic, this is a vial which will be having the 100 numbers of disc, a specific disc with a specific concentration of antibiotics. So the disc will be, you can see in a third picture, you can see here like stickers, round white color stickers you can see here, right? So that stickers will be inside this bottles. The specific antibiotics like azithromycin, nitrofurantoin, ciprofloxacin, right? Ofloxacin, tobramycin, azithromycin, right? Different types of antibiotics will be there. The antibiotics disc will be, this is a disc, small white color discs are there, no? That disc will be treated with the azithromycin, nitrofurantoin, meropenem, or norfloxacin, no? Like different types of antibiotics will be treated with a lower concentration. 10 mcg, 25 mcg, 100 mcg, 50 mcg, different concentration will be there. So that specific antibiotics for the gram positive bacteria, there will be a different antibiotics. Gram negative bacteria, there will be a different antibiotics. For the specific E. coli, Pseudomonas, Staphylococci, pyogens, Bacillus subtilis, different types of bacteria, there will be a specific chart will be there. The specific company, if it is different types of companies will be providing different antibiotics along with the chart also they will give. Okay, so the what type of bacteria, what kind of antibiotics we have to select. Okay, so what we have to do, third picture you can see. This is the plate where we have swabbed the organism. On that plate, this is the dispenser which will dispense the antibiotics evenly. If you are placing antibiotics, earlierly we have to place the antibiotic vials inside this dispenser. After the lawn culture, this is the lawn culture we have did. This is the dispenser. On a lawn culture, we have to press that dispenser and the antibiotic will dispense like this. See here, backside you can see different plates. One, two, three, four plates you can see. So the lawn culture we have did on that we have dispensed the different types of antibiotics. 8 to 10 antibiotics we can place in a one plate. That is a maximum. Okay. So the inoculum what we have to select is a McFarland standard we have to select. A McFarland is a standard turbidity we have to test. We have a hemoglobinometer. In that turbidity will be there. No, the turbidity and also test you are going to differentiate. You are going to match the turbidity with your test. Like that, the McFarland standard will be there. The organism will be inoculated in a test tube, and we have to check for the turbidity of the growth. The organism will be inoculated 8 to 10 hours, and the turbidity that will be matched with the McFarland. Then only we have to take a one lowful of organism, and we have to lawn culture this. Then we have to select the antibiotics. Then we have to dispense like this. Okay. After dispensing, we have to cover the plate. Okay, cover the plate, and we have to incubate in an incubator for 24 to 48 hours. In an incubator, 24 to 48 hours of incubation in an incubator. After the incubation, so you can see here, we have to pour the inoculum, spread the inoculum, place the antibiotic, and incubate. Okay. After incubation, how the antibiotics will show you. So here you can see in the next plate how the results will be, results of sensitivity analysis. So here you can see the picture which will be showing the different antibiotics which will be having. This is the clear zone of inhibition you can see in some antibiotics, the moderate clear zone which can be seen in the specific antibiotic, and there is no zone of inhibition in some antibiotics, right? So there is no zone of inhibition. The clear, this is what I am telling is a zone of inhibition. That is a clearance of microorganisms. So here you can see the white growth is there. No, that is a organism, and the white dots we can see the stickers. That is an antibiotic. The clear zone where the antibiotic is placed. Some of the antibiotics were showing the clear zone around the antibiotic where the organism has been died due to because of antibiotic concentration. Antibiotic concentration is there. That's why the nearby organisms have been died. So that is considered as a zone of inhibition. So that will be differentiate like a susceptible, intermediate, and also resistant. Where you cannot see the zone of inhibition, that can be considered as a resistant. Where you can see the moderate, that will be considered as a moderately sensitive, and the clear zone around the antibiotic, that will be called as a sensitive. And this zone of inhibition will be measured with the millimeters and compared with the chart. There is a specific standard chart will be available based on the what kind of antibiotics you have, which company antibiotics you are using. They will be providing the chart. You have to compare the chart with the zone of inhibition what you have got in a test, and you have to give the report in a laboratory. Okay, this is how we are going to interpret the results of sensitivity. And this sensitivity will be again same for urine, stool, sputum, and also pus. I'm not going to explain this again. Why? Because it may take more time for you people. So this sensitivity test will be again same for all urine, stool, sputum, and also pus, but the antibiotics will be varying with the different bacteria. Okay. So next we will go move on to the stool microscope. The sample will be arrived for a laboratory. So the laboratory person, what the laboratory person will do, they will check for, any ovocyst is there, or motility of organisms are there, amoebas, flagellates, if anything is there, cyst formations will be there. So for that, what the technicians will do, they will get the sample, and first and foremost thing, we have to check for the sample physical appearance, whether it is a liquidy, semi solid, or semi solid. We have to check for the consistency of the stool sample. Like that, we have to check for physical appearance of these samples. All the samples we have to do: urine, sputum, stool, pus, everything we have to check for the physical appearance. How it looks like, the color of the sample, consistency of the sample. We have to note down. So, a solid, semi-solid, or liquid consistency we have to note down. The stool microscopy will be done on a two clean glass slide. So the one glass slide will be treated with the lugulose iodine. So this is a lugulose iodine, and this is a normal saline. Okay, first what we have to do, the clean glass slide we have to take, and one loop full of samples will be placed on both the slides. One loop full of samples will be placed on both the slides, and this slide will be treated with the poured with the lugulose iodine. Lugulose iodine. So the lugulose iodine, mix it well, place a cover slip. Okay, leave the preparation for the identification. The second slide, treat with the normal saline. Place a looful of sample, mix the sample with the saline, normal saline, and place a cover slip. It should not enter any air bubbles, and leave the preparation for identification. Under the microscopy, you can see the huge, if it is any sample will be containing any protozoal motility, tropozoite motility, or cyst will be there, or amoebic or flagellates will be there, you can see under the microscopy. So different eggs, worms, and also eggs we can see under the microscopy: Giardia, Blastocystis, so Entamoeba coli, so Trichura. So all these different kinds of stool microscopic observations we can identify under the microscopy. What is the difference between this? Doing the two slides for one microscopy is this is the iodine smear, where we can see the nucleus of these worms will be get colored. Why we are doing is the nucleus of the X or cyst will be getting colored with the lugulose iodine. By observing under the microscopy with the saline, we won't get, we can't identify. Everything will looks similar. But you are treating with the lugulose iodine, the nucleus of the organisms, amoebic nucleus or some other nucleus will be get colored. That's why we are using the lugulose iodine and also saline smears. Okay, so you can see different leishmania, toxoplasma, hookworm. Okay, so all these things, eggs or worms, will be observed under the microscope. As Caricium records, all the things will be observed under the microscope. Okay, again for those tool stool microscopy. After the stool microscopy, we had to go for culturing. The stool sample will be taken on the plate, culture plate, smear with the inoculation loop, and it will be kept for the incubation. Incubation period again 24 to 48 hours. And after 48 hours of incubation, we can see this colonies as it is we have seen in a urine culture. So the specific organisms will be identified by doing a colony morphology, gram staining, or biochemical test, colony morphology. Everything we have to follow same as we have done. And also after identification of organisms, we have to give a sensitivity. Again, we have to give a sensitivity and read the results for the same for stool also. Okay, and moving on to the next one is a sputum culture and sensitivity. The sputum sample will be taken early morning. Sputum will be preferably taken for a sputum culture and also for the microscopy. The specifically will be for lung infections. We will go for this sputum microscopy or cultivation. So the consistency, color, and also physical appearance of the sputum will be counted. As I said earlier, in the urine and also stool samples, the thick purulent sputums will be there. Rusty color will be there. Cream color, greenish color sputums will be there. So that will indicate the different infections of the lung diseases. So the sputum test, how we are going to do this sputum test. Okay. So first one is a macroscopic, as I said, how it looks like, the consistency, color. Okay, and we have to put it for a microscopic observation. Microscopic observation, we can do either a gram staining or acid for staining, AFB. AFB will be preferably for Mycobacterium tuberculosis. That is in a TB patients we can prefer by seeing the microscope, the sample, sputum sample, and also we have to put it for a culture on a culture media. The culture media, the specific culture medias will be available for different types of organisms. So moving on to this, especially although this one, AFB will be counted. So the AFB will be very reliant and very fast in determining whether the patient sample will be containing Mycobacterium tuberculosis or not. AFB, that is AFB is an acid phos bacilli. Acid phos bacilli, that is a Mycobacterium tuberculosis. And that will we can give a result within 2 hours. 1 to 2 hours we can give a result, rather than waiting for a culture. It may take about 2 to 3 weeks for the growth of organism on a specific culture media. Okay, so we will learn how we are going to study, how we are going to identify the AFP, that is acid for staining of bacteria. The sputum sample, the sputum sample which will be arriving for the laboratory, that will be smeared on a glass slide, clean glass slide, and it will be dried, and then it will be placed on a saturated paper. Saturated paper will be placed on that. On that, the carbal fustin. Here we are having a three stains. That is, the first one is a carbal fustin. Second one is a decorizing agent, that is a acid alcohol. Third one is a counter stain, that is a methylene blue. Okay. Here the time also will count. That is a 5 minute, 10 to 30 minutes, and also 2 minutes. The first one is a carbal fustin that will be placed on a saturated paper. The slide will be smeared with the sputum. On that sputum, we are placing a saturated paper. That saturated paper will be applied with the carbal fustin. On the paper, we are going to apply a carbal fustin, not directly. The smear will be there. On that saturated paper will be placed. On that, we are going to pour a carbal fustin, and we are going to heat for a 5 minutes in an exhaust hood. Okay, and which it should not be, the stain should not be dried. If it is drying, we have to pour excess of stains. After that, we have to wash the slide with the water, then add a decolorizing agent, that is the alcohol, acid alcohol, and we have to leave it for, or we have to leave it for 10 to 30 seconds. Then add a counter stain with a methylene blue for about 2 minutes. For about 2 minutes, we have to leave the preparation with the methylene blue. Then we are going to rinse the water, leave the preparation for dry, then blot it and observe with the microscopy. What are all the microscopic observations we can see after doing this AFD? So here is a microscopic observation to the two picture which will reveals. The first one is a blue background with the red rods, right? So this is the AFB positive. So AFB positive, which will be giving the blue rods, the organism which will be taken up, the red color rods with a blue background. Background will be colored blue, but the organism will be taken up with the red color. Okay. Why? Because the myeolic acid, which is a cell wall composition of this AFB bacteria, AFB bacteria will be having a myeolic acid that will be taken up even after treating with the acid alcohol. Even with the treating with the acid alcohol, it won't leave the primary stain. The primary stain which we have treated is a carbal fustin that will be a red color. Even with the treatment of acid alcohol, the organism won't leave the primary stain. That much tight bonding will be there. It will be holding the primary stain, and it won't leave the acid alcohol treatment also. The primary stain won't be coming out. The last one you're treating your slide with the methylene blue. In that methylene blue, that will be taken up by the background will be colored with the methylene blue color will be there, but the organism, if it is a AFB positive, the organism will be colored the red rods. Okay, in both the slides you can see the red rods, right? So according to the National Tuberculosis and Leprosy Control Program, the AFB grading system will be like this. If you can see the 100x, the oil emulsion field length, so you can see 1 to 9 AFB seen in a 100 fields. That is the actual number. If it is no AFB in a 100 fields, that is a negative. 1 to 9 AFB in a 10 field, that will be graded as a positive. 1 to 9 AFB per a field, that will be graded 2 plus. More than 10 AFB per a field, that is a 3 plus. This is a grading according to the National Tuberculosis and Leprosy Control Program. Okay. Thank you. Thank you for everyone. I think I have covered as much as I can be. So I thank Ayurveda College and Hospital Coimbatore, Principal Sir Dr.
Thank you, ma'am.
Chandramoudishwaran, HOD and HOD of department organizing this program by HOD and Department of Yoganidana, coordinator Dr. Devi given me this good opportunity for sharing my knowledge. Thank you, everyone.
Thank you, ma'am. Thank you for such an incredible session, ma'am. Here are few questions. Can we ask, ma'am?
Yeah, definitely.
Yes, ma'am. Thank you, ma'am. Ma'am, you mentioned about the different streaking methods. So, for a particular culture, how is the streaking method decided, ma'am?
Sorry.
Am I audible,
Different?
ma'am?
Yeah, yeah. Tell me. Different
Uh,
streaking methods.
yes, ma'am. Yes, ma'am. You mentioned about different streaking methods. So, for a particular culture, how do we decide what streaking method should we use? Whether it is simple or quadrant? Which method, ma'am?
Generally, we can use zigzag and also parallel streaking methods for all types of samples, urine, stool, pus. All generally you can use both lawn or parallel streaking methods. Both will be giving fine, pure isolated columns.
So, is it depending upon the organism that we are going to culture, or on what factor will it depend
That
on?
is that is a general methods that won't depends on what type of bacteria we are going to cultivate. You can use
Okay, ma'am.
all the samples. You can use streaking methods, the zigzag method and
Okay.
also parallel method. You can use all the samples. There is no specific organism we have to do streaking like zigzag. You have to go for not like that. Nothing like that. You can use both the methods.
Okay, ma'am. Ma'am, just in case if there are more than one or two microbes which are sharing common features, so is there like how we have for diseases we have differential diagnosis? So is there any exceptional case existing in microbiology as well with respect to urine, sputum, etc. samples?
Come again. Come again.
Ma'am, just in case if more than one microorganism shares a common feature, is there diagnosis? Is there any differential diagnosis which is present, ma'am?
Come again.
Ma'am, you are not audible.
Sneha, can you hear me? Sneha?
You're not audible, ma'am.
Sneha?
Ma'am, you're not audible, ma'am.
Now?
No, ma'am. You're not audible.
Neha, ma'am is audible. She's clear.
Audible?
Yeah, you are audible, ma'am. Sneha, check your connectivity.
Sir, tell her to ask the question, sir.
Sneha, just put the question. Sneha, you are out.
Yes, ma'am. Can I proceed with the questions, ma'am?
Yes.
Okay.
Yes,
Sorry for the
Sneha.
interruption, ma'am. Yes,
Tell.
ma'am. The doubt is, in case microorganisms share a common feature, so how will we
Mm.
differentiate between the organisms, ma'am? Apart from the that is microscopic picture,
Yeah, yeah,
is
yeah.
there any way of
So,
differentiating? Yes,
Definitely. For example, you can
yes,
see here the slide what I have shown you. So this is the Enterobacteriaceae members, the Meconium agar. I told you the Enterobacteriaceae members
yes,
which will
ma'am.
be sharing the similar colonies, and also similar features, right? It might, this is the E.
Yes,
coli, this is the Pseudomonas, right? So this is the E. coli which will
yes,
be showing a lactose fermenting. This is the non-lactose fermenting which will be the showing the Pseudomonas. For example, one more example I can give you is the Klebsiella and also the E. coli, which will be sharing the similar features, colony morphology. But one of the tests what we can do is a stringe test. Stringe test is a method. If you are lifting the E. coli and also Klebsiella with the help of stick, applicator stick, you can see while lifting up the colony along with the stick, you can see one stringe like consistency. One stringe will come. That is a stringe test positive, we will call it as. That will be positive for Klebsiella, not in E. coli. That is how we are going to differentiate the colony morphology. Even though if it is sharing a similar colony morphologies, we can identify by doing some specific test like a stringe test. There is a different
okay.
methods are there for identification.
Oh, okay, ma'am. Thank you, ma'am. Ma'am, is
Okay.
it possible that multiple types of microbes are present in the same sample, and if so, how will we culture and identify them?
Yeah, definitely. There will be some cross contaminations will be there, and also with the urethra. Some will be from the, if you are taken on from the skin samples, other than a pus sample, which will be having a pathogenic organism, you may get the skin normal flora also. So by identification of colony morphology characteristics, we can identify whether the organism is pathogenic or non-pathogenic. We can identify.
Okay, ma'am.
By biochemical
Okay,
tests also
fine.
we can identify.
Okay, ma'am. Ma'am, I have heard that the most commonly used medium for culture is solid medium. Is there any like specificity? How do we decide this medium, ma'am, whether to be solid or liquid, or we go with the standard solid medium as it is more comfortable? Uh,
See, for solid media, that is a widely used media for specific organisms. You can use a liquid media also, liquid media, and also semi-solid media. As I said, semi-solid media is specifically for motility test. But solid media and liquid media, where why we are using different medias is liquid media will provide you to exhibit all kinds of features, all kinds of features. That is, a flagella can be exhibited. Flagella can be exhibited in a liquid media that may not be exhibited in a solid media. Solid media will be substrate media. That in that the
Okay.
organism may not exhibit specific characteristics that will be exhibited in a liquid media. So that's why we are using both the medias, liquid media and also solid.
Okay. Okay. Ma'am, can you throw a light on the role of microbiology with respect to oncology? I know that it is not related to the topic, but a few things about it maybe.
Okay. So the oncology, coming to the oncology, some of the microorganisms can also causes a viruses, tumors. So Epstein-Barr viruses, so different types. Am I audible?
Yes, ma'am, you are audible, ma'am.
Yeah. So by yeah, not only the tumors will be had by the um, other than the microorganisms, the various types of microorganisms which will be causing the tumors in human beings, that is very dreadful. And as I said earlier, Epstein-Barr viruses or some different types of viruses which will be causing a tumor. And the nowadays we are having we have vaccinations also for those types of tumors. The upcoming years we may expect good vaccinations for these types of tumors which will be caused with the microorganisms. And that needs a very sophisticated lab structures for studying all these things. And also we need individuals. We can't use human individuals. We have to study for, we have to sacrifice for various animals will be there. Study animals will be there. So,
Okay.
so
Yes, ma'am. Ma'am, is it possible that virus can be cultured?
yes, not on this artificial medias. There is a different cultivation method for viruses. That is, a cell culture methods will be used. Embryonated eggs cell culture will be done. Specific viruses, there is a specific cell cultivation will be done. Cell cultures will be not on this artificial medias. We can't culture the viruses on this culture medias. We need a specialized cell culture medias for viral cultivation.
Okay, ma'am. Thank you, ma'am. Uh,
Yeah. Thank you.
can we
Thank you,
move
Sneha.
ahead with the vote of thanks, ma'am? Yes, ma'am. Thank you, ma'am. Can we move ahead with the vote of thanks session, ma'am?
Definitely.
Yes. I now request Dr. Devi
Yes,
S.
yes.
Nair, ma'am, HOD. Yes, ma'am. I now request Dr. Devi S. Nair, ma'am, HOD, Department of Rog Nidana Evam Vikriti Vigyan, Ayurveda College and Hospital, Coimbatore, to deliver the formal vote of thanks.
Dr. Chandramouli. It's audible. Sneha, is it
Yes, ma'am,
audible?
you are audible, ma'am. Yes, ma'am, you are
Okay,
audible,
okay,
ma'am.
thank you. Yeah, so I would like to
Yes,
thank
ma'am.
Dr. Chandramouli Ishwaran, the principal, Ayurveda College, Coimbatore, for giving us all the required support. And this program is not possible without the hard work of Dr. Samuel. Behind the program, his good mind to help the students. So always, he's a part of this program only. Thank you so much, sir. And not at the last, actually, I should thank the speaker first. But ma'am is very special for me. She is my microbiology teacher, and she is such a efficient teacher in practical. She will ensure every student of her is able to give a report of the patient. Like that, she has taught us and very interesting practical classes. Ma'am, I'm really, really thankful that you are taking a session for my students and other students in this India. And uh, thank you for giving your time. Thank you so much, ma'am.
Thank you, ma'am.
Thank you, ma'am. So we have come to the end of the session. I would like to thank our key speaker today. Thank you, ma'am. You have made our day very special with lots of knowledge, and we hope that it will be very useful for us in the future. Thank you, ma'am. Thank
Thank
you
you
all
so
for
much.
joining us. Thank you. Yes, ma'am. Thank you.
Thank you all.